it was proposed that India is becoming vice chair, and that is it will continue for 3 years. Then India will take the lead. So we have been doing as per GLP. As I said, that we have almost 50 plus lab, which is GLP accredited lab, and that is OECD GLP. So normally when you say word GLP, good laboratory practice, we follow the good laboratory practice, but in a principle of OECD. That is what I wanted to highlight to all the participants, that OECD means Organization of Economic Cooperation and Development. And based on this principle, we have a mutual agreement, mutual acceptance of data. That is, you call it MAD. I'll just give you one example that how MAD is applicable. That suppose if you say you do a study and you send for publication, and you see that some of the journal, reputed journal, they send you back within half an hour and with a very polite language that we have a very high volume of submission of paper. So you paper did not have a priority, so we are rejecting, you submit it to another journal, or maybe in a future we'll be considering. Why it is so that within half an hour, within 1 hour, within a day, it has been rejected? The reason behind, people might think that you are working on some lab which may not be accredited or not in a global priority, so they send it back. And that was actually happening when earlier you do some of the studies in UK, and when a company come to the France, they also say that, we have to repeat the whole test because we do not accept the data of, from the UK. So that means when you do the same experiment again, that means it is going to cost you. Then you have a time, whole time, maybe preclinical toxicity study, as I say, it is around 3.5 years. So now the concept is coming that if you follow the same SOP, same principle under OECD, Organization of Economic Cooperation and Development, then the data can be accepted. So priority is that we have a GLP lab under OECD, which are 50 plus GLP lab. So if we do a study, we can go to US FDA, you can go to Europe, you can go to Japan, any place to the regulatory authority. And we say we are GLP certified lab. So that's how that we have to take a concept. Now I'm coming to the history that how GLP has been started. So if you say by definition, it is basically a quality system. Good laboratory practice is a quality system that everything you do, like you do a studies, we plan a protocol and there is a particular protocol number. So whenever you write a protocol, then there is always a version. So you say, what is your protocol? This is a title, this is a protocol number, and number itself will signify you that how many amendment has been taken place. So every amendment you go to the, Institute Ethics Committee or Ethics Committee get an approval. So we get an why you have taken an amendment. So this particular GLP is a quality system. It talks about, how you are going to plan the study, how you are going to perform the studies, how you are going to monitor, and how you are going to record and archive and report it. Now, if you ask a question, I'll ask again after the presentation. What makes you difference if you say GLP studies and non-GLP studies? You know that any accredited system, when you do the studies, there is an internal quality and external quality. So in GLP also, there is an internal quality, which is done by quality assurance team. So there is an internal audit is there. And the external audit is that there is a GLP monitoring authority. That is done by DST, Department of Science and Technology, where you have a national GLP monitoring authority. So they also accredit every, once it is approved the lab, whatever study they do, they again come back in 1.5 years and they go for a surveillance inspection. Then there is another full inspection is done after 3 years. Now, as I said, it is a quality system of the GLP under OECD. Now, how it was started, if you go back in 70s, there are so many labs in US, there are currently at that time around 40 labs. They are doing a lot of studies, maybe 20,000 studies, more than 20,000 studies. And some of the people, they could find that all the studies are very badly managed. You know that you do a studies, if you say that, why don't you come to me and see my data? That means you are confident. That means you can talk about reproducibility. That if you do the experiment again, and it is reproducible. So all these 40 laboratories, when you see that all the data were badly managed, and that time there are a couple of CROs who were doing the business since 1953. And by the time when investigation was on by some of the regulatory authority from US, Environmental Protection Agency, then US FDA, so by far they have already done more than 22,000 studies. Now, when it was investigated, they could find that all the people who had done the studies, they were not trained, neither the study director nor the study personnel. And the design was not adequate. So normally if you see that if you do the study, as I said in my lecture, that even if you are a professor, when you go to a university, you wanted to do the study, there is an orientation program. Then you also know that particular facilities, what are the SOP, standard operating procedure. So you taken training for that particular SOP also. So if you plan a protocol, if it is not properly designed, like you have a control group, you have a fecal control group, you have a positive or negative control group. So any drug which is currently in the standard therapy, you have to keep it a control group. Then only you can say that design is adequate. Otherwise, you cannot extrapolate the data. So there are so many problems because of that. And if you do an experiment, any group you keeping 6 to 10 animal, so there should be identification mark, that how you identify group number one animal, 2 animal, 3 animal in terms of body weight, then how much dose of the drug you are going to give. So if you cannot particularly identify, then it will be a misleading information you are generating. So there is no standardized procedure during that time. So you happen to go to animal house, you do not have a healthy animals, animal IDs were not maintained, especially if you say that I am doing the studies in Worcester rat or Sparkdolly rat. So do you have a characterization? So all our equipment, normally when you go to lab, if you say that it is accredited, see an instrument, you say whether there is an ID of the instrument is there or not. Is there any SOP has been displayed? Or is there any logbook? So when you see the logbook, then you ask for any calibration is required. So when the calibration is done, when there is a due for next calibration. So, and even suppose during the experimental stage, if there is an animal died, you need to go for a necropsy. That means you have to go for a histopathology. Like in human, we do postmortem. So that is what we wanted to find out during the experiment, why the animal died. What is the reason? Is it because of some, management in the animal house is wrong, or there is some other problem is there? So there could be spontaneous tumor. So all these things would be morphologically, histologically, and other method you have to explain, mechanistic exploration, or identify the root cause for any of the mortality during the experiment. So all reported animals were not examined. So that means the data were falsified, and there is no accountability for dead animals. So these all problems were there. And normally in GLP facilities right now, whenever you do a studies, we keep the data, whether it is dry, like paperwork, or you have a wet tissue, like you keep internal organs. So we keep it for 3 cycles. 3 cycle means you are going to keep it for 9 years. So during that time, we call it archive. You're going to archive all the data, raw data, for almost for 9 years. We call it 3 cycle. And wherever the sponsor is there, they do not have a monitoring mechanism for any share during that time. Then ultimately, it was decided that we should have a standardized procedure for across the globe. So in 1975, that there is an investigational industrial biotest lab. They come with a publication of GLP and FDA. FDA is Food and Drug Administration along with the EPA. Then there is a series of publications. If you look at since 1976, then 81, 83, and 1989 on GLP. So finally, there are 29 different, developed countries from North America, Europe, and specific. They form the OECD principle. OECD is, please be remember, it is Organization of Economic Cooperation and Development. So under the OECD, now we have an associate membership. All these countries, you say, India, Indonesia, or Philippines, they are also member. Brazil is also member. But, one thing is that China is still not a member. So that is why we get a lot of business that people from China, the different companies, they come and do their studies here, because they are not till now GLP certified lab. They do not have a lab. So you start from 1982 till 1997, where OECD principle revised version of GLP has been formed. So the principle based on what I said earlier, had given an example of MAD, mutual acceptance of data. So that is on based on that, any study when you do, if you do it under GLP, then this study will be accepted across the world. All the regulatory authority will accept it if you do, it is certified by OECD GLP. So mutual acceptance data talk about, how the testing will be done under the GLP principle for acute toxicity study, or for repeated dose toxicity study, or for long-term toxicity study, or for safety, pharmacological study. That also include not only pharmaceutical product, like if you do it for pesticide, or you do it for any food excipient or food product, that is also you have to do it in under GLP. So ultimately what we are trying to make it is, you wanted to make sure that whatever data you generate, it's a high quality result. That means this result, anybody can challenge, or you can say, we can go for reproducibility, okay, and comparability. So that is what that we wanted. So if you say that I have done it from the GLP lab, that means you can say, I can go to any regulatory authority, submit the data, it is acceptable. That is what we want to promote, a mutual recognition of the result. So as a scientist, we'll be happy that you are working on a GLP lab and you can send these data to any of the lab. So please go through, you can freely download from the DST Monitoring Authority, National GLP Monitoring Authority, all the document. So all this OECD principle document 1, other 10 document you can download from this. Just go to this list of the document of OECD principle. So it talk about guideline for compliance of monitoring, how monitoring mechanism is done by GLP Monitoring Authority, how the audits are performed, how external or internal audits are performed. So how the studies are, how do you make a quality assurance of GLP? Everything it has been there in a document. You can just go through and you can freely download also. Now, when you see OECD GLP principle, any of these guidelines you read, it starts with a scope. The scope is that you wanted to go for a drug development process. So they certify that based on your expertise and skill and trained manpower, they'll certify that you are certified for doing acute toxicity study, repeated dose toxicity study, or long-term toxicity study, including safety pharmacology. Some of the lab are certified because they have a chemical testing, or you have a environmental toxicity study. Like I said, you wanted to do it in honey bees, or you want to do it in earthworm, or you wanted to do it in any of the fish. So these are ecotoxicity studies. So based on the expertise, training of the manpower and facilities, you have that all these certifications are done. So the term concerning non-clinical health, what we talk about in series of toxicity studies, acute, repeated dose, or long-term toxicity study, along with environmental safety studies, everything it is certified in different labs. Now, when you look at the organization in section 2, it talk about that there is a test facility management. So they are the main person who exactly organize, all this. They appoint a study director. Then if it is a multi-centric study, there is a principal investigator in a different centers. As I said, that every accredited system, there is an internal quality. So this is what, that you have a quality assurance program is there. So they're the strong people. If you have a good quality assurance unit, depending on the facilities, how many manpower is required, who are responsible to look at that as per protocol, as per SOP, they have done the studies or not. So they have a plan in their SOP that they want to go for a facilities-based inspection audit or study-based. So there have to be that any studies you do, they have to go and observe that as per protocol, they have the study done or not. If it is not done, that means they have to inform to the test facility management. That is, you call it, refer it to TFM. Now, section 2 also talk about different facilities, like in GLP, whenever you call it test system, it referred it to an animal system, whether it is smaller animal or the higher animal. Whatever animal house you are keeping the animal, like mice, rat, or a guinea pig, rabbit, or higher animal like monkeys and dog. Now, facilities also have handling the test and reference item. We refer it to TICO, Test Item Control Organization. We refer it to TICO. So TICO, they responsible that whenever a sponsor approves for doing a studies, so they talk to the TFM, then they get that compound with certificate of analysis. Accordingly, they keep the particular test compound. Like suppose some test compound may be, hygroscopic, you put it in desiccator. Some compound may be, photosensitive, you put it in amber, and it is a special provision when you handle a photosensitive or hygroscopic compound. Or similarly, if it is a hazardous compound, then you have to have a spillage kit if there is accidental, disposal. Then you have archive facilities. Now, as you see that as we go little modernized, earlier we have an archive is only dry archive. That means we keep the paper. And also we have a wet archive because all the animal tissues and internal organ, you keep it with a formaldehyde. So now they're coming up with more than a digital one that we have a e-archive. So archive also we have a dry archive, or you have a wet archive, or you have a e-archive. Then there is a procedure, like you happen to go to any hospital when you use an injection or any of the syringe or blood stain, there is a procedure, color coding system. You call it biomedical waste disposable, commonly you call it BMW. So where to put if you want to put a paper, where to put if there is an animal tissue. So normally you have to remember that any of this animal tissue or biological fluid like blood or any of the biological fluid, you put it in a yellow polythene bag. Okay, so that you have to maintain. Mostly the facilities maybe have a chemistry lab or a biochemistry lab or histopathological lab. So normally what they do is they made an agreement with any of the vendor who collect this all the waste disposable. So there should be a record how much disposable has been generated, and there is a logbook. So we have to make an agreement with any of this vendor where they come and collect depending on the facilities, maybe twice in a day or once in a week, how much it is generated. So we have to keep the record for any of the waste disposable. Now, as I said that all the apparatus, materials, and reagents, like normally when you see a Sigma book, so one chemical may be costing 25,000, and same chemical, depending on the purity, maybe it is 1 lakh rupees. So all this detail that internationally reputed vendors, you are going to get the materials or reagent, and also apparatus should be with accredited. Okay, so we have a calibration, everything. Then test system, you may have, as I say, it referred to animal, but it could be physical, chemical, or biological. The animal system is there. Now, section 2, we talk about test and reference item that we refer it to TICO, Test Item Control Organization. So this will detail how we are going to receive this, any of this new chemical entity, how we are going to handle, how we are going to sampling, storage, and characterization. So normally we ask for detail of certificate of analysis. Then we have a SOP. These are the backbone. So normally we call that, do you have an SOP for SOP? Because you happen to go to a small facilities, if you say how many SOPs you have, maybe they'll say 50 SOPs. You go to the larger facilities, how many SOPs you have? Maybe they have 4,000 plus SOPs. So every SOP, there is a SOP for SOP. I think all the students should exercise is how you write an SOP. What do you understand by SOP for SOP? Even if you say that you have an SOP, is it a controlled document and uncontrolled document? So all SOP need to be written that who has written that SOP, who has reviewed the SOP, who has approved the SOP, what is the version of the SOP. So detailed procedure is normally, suppose you want to write an SOP for a simple instrument like a weighing machine. Okay, so you just go through that manual of this machine, the instruction from the company, based on that you write an SOP. So all details step by step. So if we're doing today any of the experiment, tomorrow somebody else is come, then they will be trained in that SOP. So ultimately result is safe. Then you talk about performance of the study, you have a protocol. So as per protocol you do, if you cannot do, there is an amendment of the protocol. Everything, there is a version is there, you can get when somebody else audit, you get an idea that is there an amendment has been taken place or not. So ultimately you are going to report the studies. So reporting is to the regulatory authority in the form of IND, what we talked about the last lectures for a phase 1 studies, and make a final report, and you certify that this is done under principle of GLP. So you are going to keep all the data storage and retention, as I say, 3 cycle. 3 cycle means there is 9 years you are going to keep all the record. And that is what mandatory for global requirement also. Now we have a provision also that in case of you are getting a data from outside India, you have right to go, like US FDA come to India for inspection because we market the product to US. Similarly, if we get a bulk from China, then you have right to go to China also. So this procedure is there for across the border also, you can go for it. So reverse guideline, it talk about that how you certify a lab that initially go for pre-inspection. So in the inspection, you started with the conference, opening conference. So you identify the TFM, what is their responsibility, you go for infrastructure, and who are the person is responsible for everything you look for, organization and personnel, that how they train all these people, you go to the biodata, you have an interaction, physical interaction with the people about their expertise, how many studies they have done. Then you check the quality assurance program there, how they do a facilities-based inspection, how frequently they do a facilities-based inspection, or how to do a study-based system like animal. Do they have a characterization where the vendor they supply, is it adequate or it is approved by competent authority? Talk about the taste and reference item, everything SOP, everything you will go for it, and then you do the study audit. So ultimately, you know that preparation of GLP inspection report, you make a summary with all detail. You say, observation of TFM. Then you have a, different facilities like TICO. Then you have an animal house, different lab. So you make the observation, and then you discuss with the responsible person, TFN and other study personnel, study director. If they agree, mutually agree, you make a observation list. Then you have an exit, and ultimately it goes to technical committee who approve from the National GLP Monitoring Authority. Ultimately, lab is certified for GLP. And this is given, the certificate is given for 3 years, provided you have a surveillance inspection in after 1.5 years. So we can have a requesting for carrying out inspection in other countries also, that we talk about, that we have a right to go. If somebody else come and market the product here, we can have a right to go to for the inspection in another countries also. So altogether, if you see what I said earlier, is GLP is nothing but a quality system of any of the organization. How you conduct a non-clinical health or environmental safety studies. So it talked about plan, how do you plan it, how you're going to perform it, how you're going to monitor it, how you're going to record it and archive it, how you are going to report to the regulatory authority. Like in India, regulatory authority is Drug Controller General of India. So we have a representation in technical committee from DCGI normally. Now, normally if you see that, what are the product you need a GLP studies? Like we have a government institute that earlier there is not GLP certified. If you say in India, government institute, Naipur Mohali is a GLP certified lab. Then we have a CDRI, Indian Institute of Toxicology. Then academic institute, if you see that SRM is GLP certified. Otherwise, rest of them are all private organized. If you see all 50s, majority of private. So we have a very few academic institute are GLP certified. So if you plan to do a pharmaceutical product, or you have a pesticide product, or you talk about a cosmetic product, veterinary drugs, or food and feed additives, or industrial chemicals, you need a GLP. GLP, you have to do the GLP studies under the OECD principle, then only you can submit to the regulatory authority. So normally when we talk about a GLP, we just maybe thinking that we are working on the lab only. So it is not restricted to the lab. It may be in a tea garden, you are using a pesticide, or at a paddy field. So it is not only restricted to the lab, it is also outside the lab where the GLP principle is applied. So purpose I have already talked about, that we want to promote the quality system, and that is what we try to have, the creation of technical barrier, because you say that your data is accepted. That means as a scientist, you have a recognition when you do, GLP. Because earlier I have given an example of a paper you send, it gets rejected within half an hour, you feel bad. Why our paper is not accepted? So GLP gives that reliability, repeatability, auditability, and as a scientist, you have a recognition all over the world. Now, one of the thing is you may not apply, because when we go for an inspection, maybe I'm a professor in PGI, I'm not going to say that it should have been like this, or scientifically it is not right when we perform an audit and inspection. We only supposed to see that as per protocol, as per SOP, they have done the studies or not. But if there is a silent of the SOP, we'll say that SOP should have been like this, but this is silent on this aspect. So you are not going to, highlight that there is a deficiency in scientific value or any set of organization. So GLP will not talk about that scientific value. You say that as per protocol or as per SOP you have done the studies or not. So ultimately you wanted to make sure that the quality of the result, reproducibility of the result, so that it is mutually accepted across the globe. Now coming to the academic institute, because most of the students are there. So even you are not GLP, what I say that many of the university, they have a kind of a lot of issues maybe there. People have getting a thesis from outside or submitting it, so they want to investigate. So that is why all this logbook and records should be there. If tomorrow somebody question, have you done the experiment? Yes, I have a data. We can show the data also. So normally when you plan in academic institute, though it is not GLP, that you can just go to principle of GLP, that how do you plan a protocol? You have a hypothesis, you design, there is adequate design, equip control, you have a written document of a protocol. So if you write a protocol, just generate the number of protocol. This is the number of protocol, and if there is a revise, you can also say revise number R1, R2 like that. Then you train for that particular, experiment. So once you train that, you have all the facilities, that whether all the instruments, there is an ID you can generate. It is well maintained. It should be calibrated and validated before you do an experiment. And that is why the SOP in the lab should be there. So you can try to make an SOP, and at the same time, that when subsequent students come, they will follow the same SOP. And if there is a requirement of amendment of the SOP, that you can also do it. So all these test systems should be appropriate, healthy, and good. And whatever reference standard, or you are working on new chemical entity, so you ask for a certificate of analysis. Do you have a certificate of analysis, or you procure from the vendors? What about purity? Okay, so ultimately, when you generate the data, it is, you can identify, or there is a reproducibility of data. So that's how traceability, everything new, and credibility of the data is required. That is why even academic institute, you can apply that as GLP. So fundamentals of GLP, you can just take it up 5 basic points, that you have a trained person, resource person, facilities is okay, all these equipment are well calibrated, you follow the guideline, and whatever quality system documentation is that you can just tell you with the raw data, with the final report. And there is also statement from quality assurance team. So that's how you say that we have, though it is not GLP, but we follow the GLP principle. So what I talk about is very much required. That is what all these institutes are doing right now, also training the people only the faculty member as well as student also. So they should have that how to do it. So this is how you plan it, that guideline and study plan, protocol, design protocol, and standard operating procedure. So characterization, if I say that characterization is that of any of the reference item or preparation of the dose or chemical analysis, everything need to be characterized. So unless you have a documentation of characterization, then you do not do it. Okay, you ask for whatever sponsor, whatever vendors they supply, you ask for all the characterization. So anything you procure, you keep the data or keep the record in a file. So this is how for characterization of animal also, as I say yesterday lecture, that if you procure an animal, then you say what is their species, do you have a vendor certificate? Okay, you include a female animal, do they have a health certificate, or is it non-pregnant? So everything should be record, should be there with the characterization. Now, some of the studies, regulatory authority, as I say, GLP is applied, but some of the studies, GLPs are not applied. But however, you need to know that ultimately when you generate the data in the GLP, we go for a IND application for first time in human, and following that phase 3 trial, you are going to apply for NDA, new drug application for marketing approval. Now, some of the studies which GLP regulation apply, I have already talked about ADME studies, all the toxicity study, whether short-term and long-term. So everything it is apply, it is. But in case of a basic research, what I'm requesting is, though it is not required GLP, please follow the principle of GLP. Like basic research, what we do, thesis, developing new technology, methodology, dose ranging studies or stability studies, we do not do. So we have to keep the key activities for GLPs, all these SOPs, standard procedures, specification, review, distribution, everything we apply for GLP. Most of the people we work is like biochemical person, pharmacologist, physiologist, microbiology. So even if you do a microbiological lab, when you do this testing, you should be familiar with what kind of biosafety level you are working at. And that is why whatever training you do, there should be pre-assessment, there should be post-assessment. So if you have an SOP that you want to train the person, and if they get more than 70%, then you accept it. So then what you can do is you make an, if those who are not getting 70%, you go for a re-evaluation or retraining it. That is how we do it. So these are some of the key activities, or adequate recording of the raw data. How do you say raw data? So all this should be there. So ultimately, if you see that in principle, that we have a test and reference item, we have a quality assurance unit. This is very important, and facilities, record and report. All these apparatus, materials, and reagents are calibrated. You have an SOP, you have a test system that is animal. So if you ask the question, what do you understand by GLP studies and non-GLP study? Simply, if you take out the quality assurance statement from a studies, it is automatically become a non-GLP studies. And that is what that training and all this adequate training and a record should be maintained with all the organization person. So we have an SD, study director, who approved the protocol. Along with multicentric study, there is a PI or study personnel, and these people are trained for this. And that's how continuous training is there. So normally when you look at the record, you keep the record that how many in-house training is there, how many external training is there. So this is very important point, as I say, GLP and non-GLP quality assurance unit. So they should be very, very trained person. And they should be very prompt to report to TFM if there is any deviation. And that's how validation of analysis, this all should be there. But at the same time, I think we have learned about personal protection during the COVID-19. Earlier, when you go to the lab and ask that what is PPE, nobody knows that. Now a common man know what is PPE, marks. Okay, so this is very important, a personal safety, that while doing the experiment, the gloves, safety shoes, apron, even those who are in a chemistry lab, they should wear the glass. Everything should be. Some people have a habit that using pipette in a mouth, these are not acceptable. Then when you say that in an experiment, whether you have a first aid box or you have an eye wash, even you say you have an eye wash, that I'm going to ask that this eye wash tank is connected to the main tank or there is a separate tank. Because many of the times, main tank is empty. If there is an emergency, then there is no water. So in case of a body wash also similarly, there should be separate tank for connecting also for personal safety. So animal facilities, I request you to visit any of the GLP lab so you get an idea that how the animal facilities, in case of particular specialized animal, how you keep it. So you should visit the animal house, that there's an air curtain, there's an entry and exit, you have a record, then you have a clean corridor, then dirty corridor, how you maintain the air pressure. So air pressure inside the room is high so that it will come out. How do you maintain the temperature? How do you maintain the humidity? And how do you maintain light and dark cycle? So this should be knowing, and how to manage in terms of case, how to handle the animals. We have already discussed about this. So regulation, we have already talked about that any experiment you do, so that it is smaller animal and higher animal, it has to be regulated by different committee that we have already discussed, and handling the reference item. So ultimately, that people should know how you document. So I just refer it to one of the picture. I think you might have seen that picture, dirty picture. So there, Bidya Balan is to ask that, somebody asked the Bidya Balan, she is the heroine of the picture, that what do you understand by a film? And she answered it that film means it is entertainment, entertainment, and entertainment. So GLP also talk about documentation, documentation, and documentation. So if you do not document, in GLP you say that some people have a habit of writing on a hand. They said, if you do that in GLP, you should cut your hand and put it in a file. So The raw data should exactly tell you that who has done it, what was done, how it was done, when it was done, and it should be, coding system to do there. So any people say that there is a software, then you are going to ask that, is there any validation of the software? So we have a lot of e-record is there, and how you have an SOP for e-record also. Okay, so any of the raw data, if you see, it should be directly, promptly, accurately, and legibly, it should be there. So you sign it, and many times you write it, there is a mistake, you just cut it, accurately you write it and sign it, that there is a writing error. So that will have to go. So ultimately you prepare the report with all these symptoms like name, address, date, like you do submit a thesis, everything you do. So you submit to regulatory authority. But while submitting, you have to keep in mind that there is a quality management, quality control, and quality assurance statements should be there. This is all the data generated as per OECD principle, and the quality assurance team say that they have an audit and it has been verified all the data as per protocol or as per SOP. So anything left to this study over, you have a study plan, raw data, everything we are going to archive it. And there is a SOP also after completion of study when you are going to archive this, all the document. There is a SOP for particular facilities, and that you have to follow it. So there could be weakness. That means GLP required a lot of manpower, skilled manpower. Maybe it is costly. But if you see that if you do the study and, you can confident that we have done the study, there is a reproducibility, and so that whatever product it come to the market, then you can think that this is a quality product and people can consume it. So we have a strength in GLP because it can be verified. You have a documentation, and it can be checked or audited by any of the expert or auditor, or maybe internal and external auditor. So regularly we do a lot of training program, and you can check our lab that we call it EPL lab. Whatever the program is there, it is displayed in a EPL website. Just go to it and let me know any interest for any training also. We'll be happy to do it. And thank you very much. And I think I have just given a brief, and there is also another guideline which called GCLP, Good Clinical Laboratory Practice, that is applied to biochemistry lab. Like we have an enable accreditation. So when you do clinical research, that need to be all the lab when you generate, it has to be GCLP certified lab or enable accredited lab. So one is GLP, which is referred to preclinical toxicity study or non-clinical toxicity study for any of the drug development process. Thank you very much. With this, I'll be happy to take any of the questions. Thank you.
Thank you, Vikas sir, for such a wonderful presentation on the very relevant topic on the GLP. Sir, I'm having one personal question, means where all these GLP guidelines and documents are available if we are in the process of making our laboratory GLP certified?
As I say, India GLP is mandated by GLP Monitoring Authority under Department of Science and Technology. Dr. Ekta Kapoor is the Vice Chair for OECD also right now. So just go to the GLP Monitoring Authority under DST. We have all the document, and you can download the document. So I request everybody to download and read it, so that even if you do academic studies, you can at least show that, okay, these are the OECD principles, we have to follow them.
Okay, and what about this EPL, your program means this is the online program?
No, EPL is my lab, Experimental Pharmacology Lab and Neurobehavioral Lab.
Okay.
We have mandate, like I look after pharmacovigilance, metrovigilance, then anti-doping program. Then we have a series of programs, like normally in a year we do for neurobehavioral program. So those who are working on the neurological area, so they can be trained. That we train the people that how you extract the animal behavior into the clinical simulation. Okay. Similarly, we have a NACERA, National Workshop on Experimental Pharmacology and Alternatives. So these are 5 GCP program. Okay. Then pharmacovigilance we do very openly, metrovigilance program. Okay, so clinical. Tomorrow I'm going to do another program for rational pharmacotherapy. So this regularly we do. So people, the student who are like to do it, they can join us also.
Right. So I would like to all the participants, if they are really willing to do these kind of program, they can directly join to the Vikas Mehndi sir for this all things. So thank you very much once again for such a wonderful eye-opening. From my point of view, this is why eye-opener presentation on the GLP guideline, the regulations. We told us that's why the GLP is required, means I personally means this is my personal point of view that this is a kind of the gray area for being an Indian, because as you told that only few laboratories are certified till now, GLP certified, and even the China are not having the enough GLP certified laboratory, and China is well
Not
doing in.
till now, not recognized by OECD. So they do not have a GLP certified lab. So we have an advantage as an Indian that we have 50 plus lab which is GLP certified.
Right, sir. Right, sir. You're
I
right.
happen to the lead inspector for Government of India as well as GLP as well as GCP and NIBH. So we go to almost all the lab for audit.
Right, sir. So thank you very much once again, sir, for this wonderful presentation. And I hope my participants will get benefited and means what are the requirement of the GLP and how means if they are in the process of making their laboratory, or if they want to work in the GLP certified laboratory, what should be the requirements. So thank you very much once again, sir, for joining us in this 5 days FDP programs. Thank you very much, sir.
Thank you. Thank you for the opportunity. Thank you very much.